next speaker is Professor Eleanor Dodson, also from York. Eleanor has been at York since 1976 and was an early member of CCP4. She was also a lead scientist and stepped down the beginning of 2020, I think. She's a fellow of the Royal Society as well, and her research areas include data processing aided by computer advances, especially involving data validation and having methods and metrics to check the large amount of structural information that's being produced, especially as every year there's more and more information being added to the protein data bank. Eleanor, as well as methods, she's also been involved with several important structural studies at York, including lots of work on various insulin proteins. Eleanor's lecture is going to discuss the problems you're likely to encounter in structural refinement and how we can ensure that the models we produce are correct given the observations we have, and how we can quantify that with a validation score. Over to you.
Okay. Do you want me to start?
Yes, please.
Okay,
You have 20
I'm
minutes.
try and do this, but I may fake a mess of it. Have I got? Okay. Here, this is going to be challenging. Let me go back. When I put this date down, January 2022, I realized it's exactly 50 years since Dorothy Hodgkin came back from talking to Lyle Jensen and saying, we must try to refine insulin. Because at that stage, we were quite satisfied you'd solved the structure. And I said, it'll be a lot of work, Dorothy. And was very reluctant to start. But anyway, here we are 50 years on, and refinement is absolutely part of every structure solution these days. And I'm sorry this is going to be boringly scientific after Kevin's talk, which should have been the last talk, and then we could have talked about it all evening. I'm back to crystallography. I ought to thank the organisers, but when I saw the invitation they sent me, which is to write a practical reflection, problems typically encountered, when to know when the automated solutions were appropriate, develop model hypotheses in 20 minutes, I thought, thank you, Rob, thank you, Melanie, thank you, Svetlana. So I'm going to fail dismally, but I'll bring up some of the questions that they've asked me to discuss. Paul Emsley kindly made this slide, pointing out that in the last 60 years, productivity in terms of structures deposited in the PDB it's exponential. The ease of use of the software has improved exponentially. The first direct methods programs involved you typing every reflection in by hand. But of course, as productivity and software becomes easier to use, the ability to you're not forced to learn what you're doing quite so well. So of course now users are not nearly so skilled probably at taking a failure and extracting information from it. So I just thought I should address some of the problems. I'm sorry, I'm a boring old lady and I lecture young people, and you're all young, I suppose. The fact that we can work at such speed means that we have to keep proper records. Not we, you. And we are very lucky in York in that we have a very authoritarian computer and software manager called Johan Turkenberg, who meticulously keeps records, and I'll comment on this later. But not all labs are like that. People send me difficult structures. There's a software package called SIMBAD, which finds out whether or not your structure has already been solved. And it's rather embarrassing how often people have submitted the wrong crystal to have the data collected. I don't know why, but it's lack of record keeping. The other thing is, in the past we had time to learn the basic crystallography, and now often you don't. And the details of the pipelines seem unimportant if you get a good answer at the end. But when the automation fails, it's hard to find where things have gone wrong. So suggest the solutions that people should think about as students is, if you have a resident lab expert, don't be embarrassed to pester them. And the bulletin board is a wonderful tool where you can say, you know, what has happened, why has it happened, and very often you'll get a helpful answer. And of course, the other problem that speed brings is that we often only begin to evaluate the experiment at the and validate the structure at the very end of the process. People are going to talk about this much more, but it's absolutely important to take the validation as seriously as we possibly can. And then just what problems can the pipelines bring? During lockdown, I've been looking at some very old problems, and you know, some of these highlight more general problems. One of the things is that space groups, people more often than like to think deposit structures in the wrong space group. And I was just talking to Robbie, he said that he found something like 1,000 structures where the symmetry was too low. It's very easy to index H32 as C2, for instance, and then you instead of having one molecule per asymmetric unit, you'll have 9, is it? Or anyway, far too many. Another thing is people, they crystallize an isomorphous structure and then they do molecular replacement. And molecular replacement will put the solution on a different origin, or it may very well, or a different symmetry mate, and it makes the comparisons very difficult. People overlook their anomalous scattering information. So there's a lot of waters which are actually calciums, and you could have detected that if you looked at an anomalous map. Kevin, sorry. When you insert residues in an automatic build, they get left in the final structure, although they obviously aren't there. People place waters too early, so rebuilding is distorted. And very, very often, the complex, if you've done molecular replacement, you've got to actually think, is there a complex? Because as far as molecular replacement occurred, 2 molecules in the right place in the crystal lattice are satisfactory, whereas in fact those 2 molecules maybe should be put together to form a dimer. And just another thing that pleases users, if you find something that's bug, feed it back right to the bulletin board and say, why has this happened? Because as developers, we absolutely depend on people reporting infelicities or errors. So that's a little homily, but nothing to do with really what I was asked to talk about. Just to recap, our aim is to understand biological functions, and we know that knowledge of the structure can help us understand that. You know, this is just a sketch of the ribosome, but these structures that were determined on the right contributed to understanding how a ribosome worked. Now, how do we as crystallographers approach the whole thing? Well, we have to express the protein, we have to crystallize it, collect the data, get some phases, build a model, refine it, validate and interpret. And so we're going from the experiments. There's a great deal after the experiment, after the data is collected, before we come to the deposition. So all this computing and structure needs to be validated. And Bayes' theorem underpins the way I think we all think about things. You know, we want to look at an experiment, we want to give a probable explanation for it, and we'll look at that experiment in light of our prior knowledge and what the experiment is telling us. And that's expressed in the Bayes theorem. Well, I'll come to that. For us, the desired result is a list of coordinates, which will give us just a snapshot of the molecule we're interested in. The experiment is going to lead to the set of Xray observations, and then the theorem can be stated as the probability that the model giving the observation is the probability that the model is a sensible piece of chemistry, the probability that the model predicts the observations, divided by the probability that the observations are correct. So all of these issues we need to worry. Are our observations reliable? Have we built a sensible model? You know, all the carbon bonds 2 angstroms, probably that's wrong. And then we're going to refine by shifting the coordinates to maximize that probability. And every probability, of course, has to come with an error of uncertainty to do it properly. So look at the probability of the model. This is going to be discussed later. We now know a lot about the chemical structure, the bond lengths, the secondary structure, and so on. They're tabulated into dictionaries, and as things stand at present, the dictionaries for the standard chemistry are very good, and the refinement procedures are going to use these estimates of error to decide whether your coordinates are sensible or not. But there are still problems. Some information is still missing. Metal, organometallic chemistry isn't very well described in the dictionaries. I think now the sugars are sorted out, but it was only 2 or 3 years ago that people began to realize that there was serious errors in the way the sugar dictionaries were put together. And our proteins have these irritating habits of forming links and sort of covalent bonds, which are also sometimes not described very well. And then, of course, the crystal environment can modify the ideal target. And also, of course, dictionary compilers can make mistakes, and especially if you're going to look at, if you have an important ligand, I think you need to read what the restraints are going to be. They're almost always right if you've given the correct smile string, but people sometimes make mistakes, and you know, Bernard Ruppel, I'm sure, will be saying how often you find that especially ligands have some very bizarre features which are probably not real at all. And that turn my attention to the probability the observations are right. This is our experiment, and I'm afraid no matter how hard it is to understand, I think if you want to use crystallography, you owe it to yourself to try and learn a little bit about diffraction. I sometimes get sent data which frankly is useless. You need to understand whether you've got something that can become a basis of a structure thing. Paul asked me to say, how does one do that? Well, I don't quite know, but anyway, other people are going to discuss that. The data is processed automatically. There's a lot of software packages that do this. They give you a likely cell. They don't give you the certain cell. They look at the duplicate measurements and so on and score how well these agree. And they provide an estimate of the error. And they all produce reports, good reports. But the problem is, do you read the reports? And if you read them, do you understand them? And if you don't understand them, do you ask somebody to explain it to you? And then something that's quite hard to do now sometimes in the synchroton, often if you look at one of the diffraction images, you can see that something was wrong. It's smeared, the spots are streaky, worth doing. And the other thing that we've just realized ourselves in the lab recently, people sometimes send 10 crystals to the synchrotron. And is there an easy way to decide which of those crystals yielded the best data? And people are looking at ways to do that now, but you do need to check. Sometimes the first data set is not the best data set that you've got. So consider the problems of the observations. Now, once you've got the model, does it agree with the observations? Well, yes, we check the R factor between the calculated structure factors and the observed, and that gives us a measure of how good it is. What's an acceptable value? If it's 55, that's random, and you probably haven't solved the structure. But Ellie's going to talk about this later. You can have solved the structure, but the absolute final value will be influenced by things like resolution, non-crystallographic symmetry, twinning, and so on. And also, you have to think, what did you want to learn? If all you want to know is it's your ligand bound, you probably don't care about the water on the far side of the crystal lattice. So we need to look at this with some consideration of the quality of the data back to the observations, and with some consideration of why we're doing the experiment. Now, this is my little hobby horse. The model is usually built in an automated way, and that automated way has been using I don't know what the time is, using all the refinement techniques, the map interpretation techniques, which all going on behind closed doors. They're automated, they're quick, and they usually very often give you a very good solution to the structure. But when it comes to the validation, the final validation, I think you really need to look at the map yourself. First of all, because you might have overlooked things, I'll show you 2 examples in a minute. And secondly, because as crystallographers, you have these wonderful things called difference maps, which will show us where things are wrong and will show us how things ought to have been corrected. So I'm just going to put up a few examples from Qt, because first of all, because I love using it, and because it's got very good validation tools built into it. And it's, that's as far as I'm concerned, that's a high point of doing a structure. Oh, and I always have to put this in, because this is Guy Dodson and Dorothy Hodgkin with the insulin maps, and these pieces of paper scattered all over the desk are the actual maps. So, things have moved on since then, when you had to peer at a piece of map and make sure you had the right section before you built a water. But it certainly made them happy. So, and now we can be happy with our Qt maps. Just quickly, I'm sure you're all familiar with this. Qt gives you a validation graph which shows residues which are not fitting the density very well. And when you look at the maps, here's an example which should really have been fixed by the automated procedures, but it's very obvious what was wrong. The wrong rotamer for this tryptophan was chosen in the first place. Then the automated procedures put a water there, and so the poor old program says, oh, I can't move the tryptophan into this, although it's obvious that the water's wrong and the tryptophan should be changed, the rotamer should be changed. So there's something which I'm sure 5 years down the line you probably won't see, because the automation procedures will have solved that problem. But it's easy to see in the map what's wrong. And here's another example.
You have 5 minutes left.
Okay, I've nearly finished. This is a harder one to deal with. Here's a case where the peptide should flip here, and it's a cis peptide, not a gly. But and if you look at the next slide, having flipped the peptide and just done some real space refinement, everything's fitting into this difference density very nicely. But here's a case where you simply could never have done it by automation. This is a structure we solved just recently, where the data was collected in 1999. It had been deemed insoluble. It did come out. Lockdown contributes. And when you look at the map, there's this interesting green blob. And with a bit of going back to the person who collected the data in 1999, remembered that often there were N-acetyl serines associated with this structure, and indeed it fits beautifully. But that required personal information and personal input. So you'll never get to I don't think ever to the end by automation alone. And just to stop, sorry, the acknowledgements are inadequate. Paul's helped enormously with the figures, which I'm very, very grateful, Paul, if you're listening. I've worked with a lot of crystallographers over the years and thought a lot about a refinement of this over the years. And since jolly old Dorothy asking me in '72 to get on with it, and just I don't know why I couldn't change the background of that to another colour, but there's been so many people contributed algorithms. Garib, of course, refinement. Paul, who's done a lot of real space, Kevin, Early, many, many more people than I've probably put up. So that's the end, and thank you very much for listening. Gosh, that was a race. So, I don't know what I've done now. I can stop sharing the screen. Yes, thank you. Yes.
Thank you for that interesting talk on problems with refinement. Time for some questions briefly, I think.
Yeah, so there's a question from David Aragao. So he says, with regard to dictionaries, I recall a discussion initiated by James Holton on the bulletin board about different refinement programs using different dictionary targets, giving results that are inconsistent, and at times, meaning large numbers of bonds or residues, seem to have small errors that built up, creating strain. Do you envisage that we should move to some kind of molecular dynamics or quantum methods at this stage?
I don't know, because I think I can't say I'm an expert in molecular dynamics, but there are unseen factors. One of the obvious unseen factors is we don't take into account interactions between one molecule and another in the crystal lattice, and almost all molecular dynamics are done in infinite solvents. So I think our restraints need to be not lies, not down lies, but as long as we've got a realistic estimate of the error, they should be good enough to let us get through with the refinement. And ideally, we want the experiment to speak to us, not the restraints. So that's my answer to that, I guess. Yes.
So I don't think there are any more questions right now in the Q&A. I don't know if there are any based on Slack.
All those points I've raised are going to be discussed again in more detail, so that there'll be time to discuss the issues.
Yeah, I guess maybe before we move on to the next talk, I mean, I think, I don't know, you highlighted so many of the pitfalls that people can fall into, and how much help there is available, if you crowdsource from your community. But I guess maybe sort of a comment is there's maybe plenty to be said for just plowing ahead and just giving things a go, right? That people shouldn't be too worried about failing with their initial analysis, because they can always sort of go back and get it right.
Yes, well, that was certainly true with that 1999 date. Surprising that anyone remembered about the ineacetyl serine. But yes, I think there are things. I know, because I've come through this old school, I do think people should try to understand their data. If you've collected lousy data, there are lots of signals in the processing to tell you that. And so often you see people have struggled on for months. In fact, that they had got a lousy experiment and it was time to go back and do it again or rethink. And then the validation tools are getting better and better all the time, and they are interesting to look at. So, and the automated pipelines are getting better and better all the time, of course, too.
So we have a question from Rakesh. So he or she says, great talk. Often when we solve structures, the density in the loop regions are incomplete. And so the question is it wise to put polyalanine in them or the actual sequence?
I think if you've got no density, you shouldn't build a structure. We haven't got a crystal solution. We know the residues are there, and but we can't tell you where they are. And you sort of lie to the community if you say you can, but other people have different views. It's an endless discussion on the bulletin board about whether you should. Yes, and luckily, the B factors speak to us. If you see a loop with B factors and the temperatures have gone up to 200, probably it wasn't very clear in the first place. Yes. And one of our problems as crystallographers is to educate the bioinformatics people to take our B factors seriously, and realize that some parts of the information we're giving them are sound and some parts are not.
Thank you. So then there's another question from Felix Veroni. What's your advice when you're dealing with small ligands like nitrite or nitric oxide?
Oh dear. Oh, don't ask.
It's a tricky one, isn't it? What do you do with that?
If you've got 1 angstrom data, you've got no problem. So, I suppose my advice is, I don't know what you do when you've got low resolution, and you simply have to say in your paper, I have reason to believe that it might be nitric oxide, and there is some evidence that I can see a blob. I don't think you'd word it quite like that, but but if you've got high resolution, obviously you can begin to say, well, I had nitric oxide and it was in 3 different conformations. So we've got to realize that you can only extract a certain amount of information from a not very definitive experiment. So I guess that's my answer. I mean, nitric oxide, you'll have a dictionary from small molecules that you can just use. Yeah.
Right. So I think we should probably move on to the next talk, and I'll hand over to Bayes to introduce the next speaker.